We are now on to the next slide, next PowerPoint. How is everything? The test there next week. Honestly, there's no surprises. It'll be exactly the same as what we've done. It'll just be a little bigger, just because we have an extra week going on in there. The nervous assignment, those should be coming in there today, and give me a day or two. I'm going to get on those because I really don't want things to slack off. So those, hopefully they should be done. I'm hoping they're by Friday. And then other than that, I don't really see any issues. I hope there's not. And if there is, let me know. If you're struggling on anything, keep me posted. But I think you guys are just right on track. Okay, and I want to see everyone get out of here with credit. I don't want to see anyone there. If anyone's having trouble or if they're fearful that they may not get credit, come and see me. We'll get you guys through, and you guys are in good hands. Any questions? Any concerns? Anything at all? I'll post there the answers there to the nervous system. I don't know when marks are posted. And because nervous system assignments should be in there today by midnight, somewhere in there. I think that's what it is. And so I'll post the nervous assignment answers there in a couple of days. And other than that, let me know if you guys have any questions. Other than that, let's go. Yeah. Okay. So this PowerPoint here it's going to be kind of, I'm not going to say hit and miss. It's the wrong complete word. But this goes kind of beyond where I want to go into prenatal development. Like, you guys are going to be PSWs, personal support workers, and you guys are going to be nurses and, you know what I mean, nurse practitioners in a couple of years anyway. So I love it. Generally, where you guys are going to be going is into, an older person's care home, but they do have younger people care homes, and you could even, I know a PSW that is taking care of, beautiful kid, man, beautiful kid, 11 years old, quadriplegic. And just the smart as a whip kid, and just needs assistance here and there. You can even get gigs being a child's companion at school, where, you know what I mean, they interact and play, and they're playing dodgeball and whatever kids do at school, and you're along with them. So that's fun. That's actually a great job helping somebody, and you're at school all day anyway. And hopefully there you guys need a reference, come and see me. We'll get you guys going to where you guys need to go. But the first half of this is on prenatal development to some extent. I'll kind of tell you what is keywords and what's not. And yeah, let's go. Because you see, we're going to describe the process of fertilization, which we've kind of done already, when, where, and how it occurs. We're going to go into a little bit more detail. Describe the process, development, cleavage, growth, morphogenesis, differentiation. Explain 3 different periods of prenatal development: embryonic, early embryonic, fetal periods. Again, you know what I mean? We're not going to go into any type of really any detail. Okay, because I don't think that this is an aspect that is going to really concern you for getting a job and moving on. Okay, but knowledge is power. So what's the harm? There is no harm. We'll state 2 functions there of the placenta. So fertilization, sperm and egg unite. We got that. When near the mid-cycle, after ovulation there, where about the first third of the fallopian tube, that's where we like it. Okay, the sperm have traveled up, and the egg is now changed into a zygote, pretty much in the first third there of the fallopian tube. Okay, and the result is what we call a zygote. Okay, and you'll remember there that 23 chromosomes come from the male, 23 chromosomes come from the female, which now makes 46 chromosomes. All right, and that's what we needed to know. All right. Prenatal development, there are 3 periods. There's the early embryonic, which is the first 2 weeks. The embryonic, which all comes from embryo, right? Okay. Weeks 3 to 8. And you can go on YouTube and you can see the embryology. That's a whole nother type of medicine. And it's not my forte at all. I will be the first to admit that for sure. But it's interesting. You go on YouTube, go on Google, and you'll see there that if you compare embryos of a human and a chicken and a dog, you'll see crazy similarities. Interesting stuff. And this is a part of science there that we are still learning about. Okay. And then you have the fetal period, which is week 9 to birth. Okay. Pregnancy consists of about 38 weeks, and it's pretty on target. About 9 months. That's what it is. And it's divided into trimesters. First, second, and third. Okay. The early embryonic period is in the first 2 weeks of pregnancy, and what goes on here, I'm just reading here from the bottom here, moving the zygote, okay, to the late blastocyst stage and implantation. Okay, but we'll get there. Prenatal development includes cleavage. Okay, maybe I need to bold type that. Cleavage, cell division by mitosis. So what are we doing here? Mitosis, we are making 2 exact copies from this one. You have a parent and 2 daughter cells. That's what they call it. You call them sons. Call them best friends. I don't care what you call them, but they're exact copies of this guy. That's what mitosis is. Okay, so it's cleavage. It's 1 cell, which then divides into 2, which then divides into 4, which then divides into 8, 16, and 32. Growth. The size of the cells will increase. Morphogenesis. Great word. Shaping of the cell clusters. Differentiation. Cells become differentiated. Like, we all have these stem cells there, and that's the new science, right? Is getting these stem cells there, that these cells, we spoke about this there a couple weeks ago, that they have the ability, they can morph into, bone or nerve or whatever you want, right? And the big problem is nerve. We don't grow nerve cells back, right? Once it's, that's it. But they're starting. There's science coming. Early embryonic period, you'll see here you've got A, B, and C, and D. So we're going to kind of go in this way here, and there's a lot going on. All right. So you got the zygote. All right. The egg, the sperm right here, they come together. There's your zygote. Okay. And then we've got what goes on here. You'll see doubling to 2, to 4, to 8. Okay. Blastomeres. That's what we call these ones right here. Blastomere, blastomere. Blastomere, 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 blastomere. Okay? So that's what's going on here. Cells are starting to double. Double, double, double. And then we've got right here at 16 cells. So it went from 1 to 2 to 4 to 8 to 16. Sorry, I don't have another 2 hands. All right? Now it is officially called a marula, because I guess that's some berry or something. Yeah, I think it's in England there. They have a raspberry or something like that they call a marula. Anyway, because it kind of looks like one. I don't see it. All right. So, and that's what's going on here. Okay, and so now we've got this marula, and now you'll see what's going on here, what's called the trophoblastic cells. And you'll see that we're starting to get into implantation. And I'm going to zoom in. These slides from this company, they're not good. Like, yeah, I can kind of see, but I'm going to zoom in here just to kind of show you what's happening. So here's your zygote, all right? And see, right in here, okay, we've got fertilization. Remember the first third of the fallopian tube? That's where we like our fertilization, okay? And then in this point here along as the zygote is traveling back towards the uterus there for implantation, okay, we get doubling of these cells, right? And then right about in here, this is at 16. It is now officially called a morula. And then it starts to get itself ready there for implantation, okay, through this phase right here, the trophoblastic phase. So there's your early embryonic. Not too much there. Do you have to memorize it? Not even close. You just got to know where to find it. And like for this test here, because there's more info on it, there's more PowerPoints, maybe what you want to do is, you know, I know you guys have all got them on a folder or something in your computer, but you know, maybe you want to make a test 3 folder and put weeks, I don't know, weeks 10, 11, and 12 into this folder. So everything there you need for this test is in this folder. Have it on your desktop so that you're going to see a question on this. Morula, how many cells are in a morula? You know what I mean? You don't need to know it. You just know where early embryonic period is, and you go to slide 6 and you'll see 16 cells. Okay, so that's what we're talking about here. Twins. The whole idea of what is a twin, right? Because the egg here, one sperm comes in, okay, this guy here eventually becomes morula and implantation. This represents one human being. Well, then how do we get twins? I think that there was a woman in, where was it? It just happened recently. Was it India or was it Africa or somewhere? Some woman had 10 babies at one time. I think it was a new record. And just the whole idea there that they have with these pregnancy chemicals, you know, to induce pregnancy. And yeah, totally. Was it South Africa? Thanks, Lynette. And yeah, you people are having. I also read somewhere that men's or male sperm counts have gone down 50% in 50 years. And the ability for females to get pregnant has also gone down as well. So something is happening with our lifestyle that is affecting our ability to reproduce. And I don't know, you know what I mean? You got to think, you know, if you're drinking water out of a plastic bottle, you ever heard of microplastics? They're everywhere, and chemicals are everywhere. People like the whole idea of having household cleaners under their kitchen sink, you know, and they clean, lemon pledge and all these kinds of things. Well, I tell my students, and you guys are still my students for another week, and I tell my students there that, okay, if you clean your whole house with lemon pledge and pine sol and all these kinds of things, and you just cleaned it all out, and the whole place just smells like a fake lemon chemical, just the way you like it, would you take a hot dog, put it on a string, and drag it through your whole house? And then would you eat the hot dog? Well, of course you wouldn't, because it has fake chemical lemon on it and you just dragged it through your whole house. There's no way I would ever eat that hot dog. And you're probably right. Okay? And so that hot dog is not clean because you won't eat it because it's got chemicals on it. But if you take a hot dog, put it on a string, and walk it through your backyard, through your back garden, and all this, could you eat that hot dog? Sure you could. A little soil never hurt anybody. Okay, it's not the greatest thing, but that actually might even boost your immune system. So just our whole idea of what is and what isn't has been shaped by advertisers. Just be careful of mutagens. Be careful of chemicals. Okay, microplastics for sure. Twins. There's a couple different kinds of twins here. Identical is the easier word. Monozygotic. So you have two twins, okay, that were one zygote, okay, 23 chromosomes. 23 chromosomes came together. We made ourselves a zygote. But then all of a sudden, it just split. And now these guys double, quadruple, 8, marula, okay? And you have two human beings that are exactly the same identically. Right down to the DNA. One person killed another person. The DNA evidence said you did it. Well, I've got a brother. Okay? So those are called monozygotic. And you can imagine, I'll throw a word out there. What does monozygotic mean? There you go. There's a question. Oops, did I just say that? Oh my goodness. Dizygotic, we also call this a fraternal. So this is where mom has, instead of releasing one egg, mom released 2 eggs. And dad, he's got 1 million sperm going in there anyway. So there you go. Now you have 2 zygotes. Now the first zygote, it had 23 chromosomes. Dad, 23 chromosomes. There's one zygote. The other one, 23 different well, I shouldn't say different, but altered, not the same, and 23 not the same chromosomes. Little changes, little differences. So now you have 2 completely different human beings. Okay, so that's that. Here's a little schematic. Fraternal twins, identical twins. And did you even know, I got this from BBC. So this, I forget when I stole this, but this counts as a reference. And this is just new. This was, I think I got this there from BBC there last year or something like that. So it's pretty recent. There's also called semi-identical twins. 2 sperm fertilize 1 egg, which divides and makes 2 babies. DNA 100% maternal, 78 paternal. Interesting, eh? So, yeah, there's lots going on out there. Here we're going to get into a little bit of embryology there. You'll see where we're going. Again, this is not my forte. I'm not going to go heavy on this at all on the test. Okay? I'll tell you where I'm going to go on this. All right? The embryonic period, okay? There's words here that I want you to know. Amnion, amniotic sac, amniotic fluid. We have an idea of what that is. Chorion, okay? Chorionic villi. Villi are those ones there that increase the surface area of a small space, right? Like the inside of the small intestine. The placenta, okay? These are good ones. There's a yolk sac, just like with any egg. Okay, the red blood cells, immature sex cells. Allantoes, we've got urinary bladder, umbilical cord. I'm more interested there in these 2 here. Okay, but you guys can have a look at this and you guys can pick, and it's all right here anyway. Okay. What I've done there is that there was some good reading in the below section here. What I've done is I've taken the same slide and I've just jacked it all up here. Okay, so apologies for reading this. The developing baby floats in amniotic fluid formed by the amnion. The baby's urine also helps create much of the amniotic fluid. Interesting enough, eh? At terms, there is about 1 liter of amniotic fluid. This protects the embryo, which also drinks and digests the fluid. The embryo secretes waste and cells in the amniotic fluid. And this is the basis there for the diagnostic test called amniocentesis. That's what I want. Okay? You should know what that is. And rupturing of the bag of water, rupturing of the amniotic fluid signals the onset of labor, generally. Okay. At birth, the membranes are expelled with the placenta as the afterbirth. We'll get there. Okay. The whole idea there of amniocentesis, chorionic villi sampling, diagnostic tests for birth defects. Now, whatever your feelings are about this is, you know what I mean. We do tests to determine if there is any type of serious illness there that cannot be rectified, and some people choose to know, some people don't choose to know. We didn't there with ours, and and I mean, just it is what it is, right? You love them just the same. So, and you'll have a look here. What they do, they take a needle, and I'll try and zoom in because these PowerPoint slides are crap. And see, just get a needle in there, no harm to baby, no harm to mama, and they draw some of the fluid out, and they can do tests on this. Because as we said, the baby's peeing in here, the baby's, you know what I mean? This is the baby's egg, yolk sac, right in here. Everything's going on in here. So we can tell a lot by the amniotic fluid. The chorionic villi sampling, let's see if I can zoom in a bit. And, okay. Now what we're doing here is we're sampling the blood in the chorionic villi. Okay, and is there a shot right here? Yep. Okay, you see what's going on in here through the vagina, the cervix right there, and just to the side of the uterine wall here, there. Okay, you'll see the chorionic villi, and then the screen jumps up here to greater magnification. And this is where we're getting in right here. We're just taking a little bit of blood right out of here because this blood is very specific to baby. Okay. Here's another shot. The placenta formed there when the chorionic villi penetrates the endometrium. Okay. So when this chorionic villi penetrates there, now we've got the start of the placenta. Okay. Site for fetal eating, breathing, excreting, secretes hormones, umbilical cord joins the fetus and the placenta. It's all happening right here. So what they do is they'll take a sample of the amniotic fluid. They'll take a sample there of the chorionic, the blood there from the chorionic villi, and they can tell exactly what's going on. Okay. And again, what I did was I took the same slide. There's some good information here, which I then just put onto here. The finger-like chorionic villi penetrate the endometrium. Okay, they sit in blood-filled spaces called lacunae, which you'll remember, that's the same word there that we were talking about for the fingernails and toenails. Same name, different function. The placental membrane ensures that the maternal and embryonic blood supplies are separate. Okay, importante. The umbilical cord connects the embryo there to the placenta. The cord's blood vessels carry unoxygenated blood from the embryo to the placenta, where it is oxygenated and returned back to the embryo. So isn't that interesting? Let's read that again. Okay, the baby's breathing. Okay, it sends its unoxygenated, so high in carbon dioxide blood, to the placenta, where the placenta kind of acts like the lungs, the external respiration. The placenta is doing that for baby. And meanwhile, let's not forget about mom. You know what I mean? She's up there somewhere, eating for two, working for two, sleeping for two, doing all these kinds of things, right? Breathing for two. She's really carrying the load. So it's, yep, good stuff. Function of the placenta, two important roles, okay? The site across which nutrients and waste are exchanged between baby and mom, okay? And it actually acts as a gland. Now, you'll know what a gland is, right? It secretes hormones that help maintain the pregnancy, prepare the body for the birthing process, and promotes postnatal events such as breastfeeding. Okay, so 2 important roles there for the placenta. The couple of words here. Genesis just means beginning, right? So here we have organogenesis, formation of organs in baby. The embryonic disc, formed by blastocyst inner cell mass. Inner cell mass forms 3 primary germ cells. These ones are good. You have the ectoderm, the mesoderm, and the endoderm. Okay, there's a movie there. Y'all can hit that. Okay, and there's good stuff down here. The words that characterize the embryonic period is organogenesis. Okay, and by week 8, the main internal organs have been established. So this is by week 8. Everything should be almost up and running. Okay. All the organs and tissues of the body develop from 3 main germ layers. There's your ectoderm, gives rise to the nervous system. Okay. Special senses and the skin. The mesoderm creates muscle, bone, blood, and cardiovascular structures. The endoderm gives rise to the lining of the digestive tract, respiratory tract, and parts of the urinary tract. I'm just reading this there from the bottom. This is good stuff right in here. Okay? And at this point in time there, the embryo weighs about 1 gram, is about 1 inch long. These guys here, teratogens, these are, I don't want to say mutagens, chemicals there that can cause mutations, but let's call it that just for all intents and purposes. These are things that are not supposed to be there, okay? And but they are. And if they are there during pregnancy, then there's a greater chance that there's going to be a birth defect of some kind, okay? Drugs during pregnancy, okay? Even cigarettes, alcohol, okay? We've got fetal alcohol syndrome. If there is, I've read studies there. A half a glass of wine 6 months in isn't bad. Don't take my word for it, but I think where the big problem is binge drinking. I think it's more than a couple of drinks at any given point in time. You get what's called FAS or FAE, fetal alcohol effects or fetal alcohol syndrome. And it's a spectrum disorder where it's not just the same in every case. There could be mild effects, medium effects, and really bad effects, okay, and this kind of stuff. But these are called teratogens, drugs, alcohol, radiation for sure, German measles. This one here, people my age, mid-40s, you might be familiar with this. Even if you're not, if you're younger, you should be familiar with this one. This chemical here was given to pregnant mothers, I think it was in the 60s, might have been late 50s, early 60s, and they would give thalidomide to pregnant females to help them with morning sickness. And the effects were just bad. Babies were born without arms and legs and these kinds of things, and misshapen digits and all this kind of stuff. So they immediately stopped that. But there are adults now who still have the effects there of thalidomide, and they're living a happy, wonderful life. And they just something really, really went wrong if you give thalidomide there during pregnancy. It's sorry, I'm just going to look down here. There's your term for teratogen, derives from meaning monster producing. Okay, You guys can have a look at these. Read up on thalidomide. It's been well documented, and there are documentaries on YouTube. There's documentaries there on Learning Channel. It was a part of history that is still around there today. But they're actually coming back and starting to look at thalidomide again, had nothing to do with pregnancy, but as a cancer-killing agent in functioning adults and these kinds of things. So there's still hope there that maybe there's some type of good that can come from thalidomide. The fetal period here, and again, we're just kind of throwing a stone against the water and just kind of skipping here. You'll, have a look. Google, you know what I mean, fetus, a human fetus at 6 weeks, a dog fetus, a chicken fetus, you know what I mean, and you'll see that evolutionarily, we kind of look the same. All of us do when we're all 6 weeks old. And you'll see there that the fetus is viable at around 20 weeks, which means that it's able to live. You know, what do they call those? Preemies, premature babies, right? So that's that. And just good to note that when a baby is... Us as adults, I think our head is about 1/6 to 1/8 of our entire body length. But when a baby's born, their head is 1/3 of their body length. So that's just like our head as an adult being this big, right? And just interesting there, the whole things that go on there in the third trimester. So the fetal period here, this is all about terms. You could imagine there might be one of these there on the test. Quickening, movement of the fetus felt by the mother. So kicking, all right. Lanugo, fine hair covering the fetus. Okay. Vernix caseosa, white cheese-like substance protecting the fetal skin from amniotic fluid. You'll see that there on the baby when he's born. And the abortion there, the loss of fetus at any time. Okay? Spontaneous, induced, and therapeutic. Okay? We're going to talk a little bit about hormone changes during pregnancy, which we kind of did there last time. Okay? And there's a couple slides there from the previous slide. You know, the wall of the endometrium building, and then you have the hormones. Know that one. That one's a good one because that's important. For your own chemistry, for your partner's chemistry, for science, for where you guys are going, okay? And because I'll ask a couple of questions from that. We'll describe the hormonal changes and stages of labor. Describe immediate postnatal changes, lifelong developmental changes. We'll hit a couple of those. And then we're going to get into hereditariness, the passing of DNA from parent to child, what's going on here. Here's one there called a karyotype, but we'll get to all these kinds of stuff there. Maternal changes in pregnancy. The rate of metabolism increases. Well, sure it does, because mom has another human being inside of her. So things are moving, and they're moving faster. Mom's eating more. Blood volume increases by 50%. So what? We have about 5 liters of blood in us. Now we're into about 7 liters of blood. Respiratory activity increases. Mom's breathing for 2. Kidneys produce more urine. Okay? And also there's that fact as well. Mom was going to pee more because as the baby grows, there's less room for the bladder, right? Nutritional needs increase. Pickles and ice cream. And I can say this because my wife did it. She was all over that. And yeah, it's actually not bad. I like sweet and salty. There you go, man. Increased hormonal secretion. Okay, we got human growth hormone, estrogen, progesterone, prolactin, oxytocin, prostaglandins, thyroid hormones, aldosterone. Everything is more. Okay? you remember their feedback and negative feedback, right? Where if we need more of something, right? Okay, the body's going to start producing more of it, but then eventually, because there's so much of everything there that we produced, it actually turns off the system. And then it drops back down again, and then when it gets too low, the system turns back on, right? That's your negative feedback. It's the body's way of always being in check. You got your baroreceptors there in your aorta, your chemoreceptors. Okay, everything is checking. Okay, it got too much. Slow it down. Okay, that's the body's way of being evolutionarily in tune. But there's another kind of feedback called a positive feedback. And we'll get into this there when we talk about labor. And you guys kind of already have seen it there with oxytocin inducing labor. We'll get there. Just be, I will ask you something on the test. Write this one down about a positive feedback. Okay? Negative feedback and positive feedback. I'd like you to know, I'm going to give you an example there today of a positive feedback relating there to labor. Okay? But know the difference there between negative feedback and positive feedback. Okay. Very good. Oh, here we are. Labor, birth of baby. Forceful contractions, they expel the fetus from the uterus. Boy, that was very scientific and it sounded horrible. We don't forcefully expel the fetus from, but I guess that's what it is, right? And the hormones involved, okay, there's your progesterone there from your corpus luteus. Okay, quiets the uterine contractions before labor. Estrogen, okay, sensitizes the uterus there to oxytocin. And oxytocin comes from the back of the pituitary. Okay? And what oxytocin does is goes right down to the uterus, to the myometrium. You remember the meat, the muscle of the uterus, right? And starts to stimulate it. And now the uteral muscles there, they start to move. And now we start to get contraction. And then we get more oxytocin. So this is more and more. And we get more muscle movement, more muscle movement. Baby comes out. The whole thing shuts off. So that is what we call a positive feedback, where it goes above and beyond and more until the job is done, and then it all goes away. Okay? Negative feedback is back and forth like a seesaw. Okay? Or I don't know what else you call it, right? It's always like this, but a positive feedback is more for one specific purpose, and once that purpose is done, the whole house of cards falls down. And that's it, it's over. So that's the difference between a positive feedback and a negative feedback. Okay? If you just go back to blood or respiration or somewhere in there, you'll find lots of examples of negative feedback or a quick Google search. That's what we're looking for. Okay? So oxytocin stimulates the uterine contractions until baby comes, and then it turns the whole thing off. Postnatal adjustments. Breathing is established. There's a whole bunch of, I think there's 12, or is it more? Or is it, there's a whole bunch of checklists there. When a baby is born, the umbilical cord is snipped. They check all of these different things. I'll write this one down here. See if I can, I'll stop sharing for 2 seconds so that I can get, if I can write this in here. I'm going to write it here. It's called the Babinski reflex. I think that's what it is. Babinski reflex. And that's where when baby's born, you know, he's got his two cute, she has their two cute little feet, you know what I mean? They're like this big. And they'll take a pen or some type of instrument and just kind of whoop right up from heel to toe. You're not hurting, but what happens is there's a very definite thing that happens. And I'll let you guys figure that out. You guys can do a Google search of that. And if baby's okay, that's what you'll get. If baby is not okay, then you do the Babinski reflex and you don't get the anticipated results, okay, hey, everyone get back here. We've got an issue. Okay, and these are these steps there that they do to make sure that baby's okay. So, breathing is established. Blood flows through the lungs there for the first time. Mechanism there for temperature regulation adjusts. Now baby's kind of on their own, right? A functioning digestive system produces meconium there, baby's first stool. Okay, green color, lots of good stuff. If you have kids, you know what I'm talking about. And most organs begin to function more efficiently there. Okay. The developmental stage, lifespan, neonatal period there, birth to about 4 weeks, infancy, 2nd to 12th, that's a tough one to say, month. Childhood, 2nd year to puberty. Adolescence, puberty to adulthood. Adulthood is adolescence to old age. And senescence, old age to death. Okay? That's kind of where we're at. Like, you know, like I could spend years on these, I don't know, what is that, 20 slides? But that's where we're at. And again, I'm not going to focus too much on these. You guys can pick out the words there. I think I've been pretty explicit about where I want to focus on these guys here, on this first half of this PowerPoint, okay? Don't go too crazy on it, but there are good words in here that you'll need. Okay, I missed that one. What else we got here? Good. Now we're into, these are the last 20 slides of the whole course. That's all right. You can scream, you can shout, smile, go for it. Woo! There you go. There you go. Come on. Is that it? You guys sleeping? All right. Let's get into it here. Hereditary. And what we're talking about here is just transmitting traits, evolutionary traits being passed on to the next generation, parent to child. And we're learning a lot about these kinds of things, like crows are passing on the ability there to recognize enemies there to their babies. You know, like the we were watching there on the National Geographic, some antelope out on the Serengeti plains of Africa was giving birth. And, you know, baby gazelle dropped down to the ground. Mom was pulling off the amniotic sac there. You know what I mean? It's a baby and mom are at their most vulnerable because there's lions, there's jackals, you know what I mean? And they, sadly enough, man, they got babies are the easiest kill, right? Out on the in the wild. But this baby gazelle was on its feet and running within a minute. And I mean running like hell. And these are genetic traits that have been passed down, mutations that have happened over millions of years, and babies who have this ability to get up and run after a minute, those babies live. The babies who did not have that trait did not make it. You know, Darwinism, the strongest survive. And this is all has to do with passing on genetic information to your next of kin. DNA, location of genetic information, blah, blah, blah. You guys know that. Chromosomes, you guys are good on those. Okay, and within the DNA, like the DNA, these strands of these chemical sequences, that's all they are. And within these chemical sequences, like G, C, 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 T, A, that could be a gene. That could be a gene there that the body transcribes, makes a protein from it, and that's a protein there that will make this organism live long enough to pass on its genes there to somebody else. Job done. Right? Who's in control? The brain or the gonads? So here's your DNA. Of course, you guys have seen this. I'll zoom in just for a little bit of a refresher. Okay, you guys remember? Can you believe we actually did the cell? Feels like a million years ago, doesn't it? It's all good. It's still in there, by the way. Don't think that you've forgotten it. Oh, it is. It is. Familiarity is your biggest friend when it comes to these courses. Because the information there that I've given you, you don't think you've retained it all. But you've got it in your bag of tricks, and your next course there, it'll still be there. Trust me. So there's your DNA. They're your chromosomes, right? They're kind of like these woolly thread-like structures there that house the DNA. Okay? And here's your DNA right here. Okay? And within the DNA, there are genes that your body will read and make proteins. We've done that kind of stuff. There are genes for eye color, hair color, style of hair, height, weight, all these kinds of things. So we're going to talk about these. I'm sure you guys must have heard of, somewhere along the line, of dominant traits and recessive traits. Okay? You will want to write those ones down. Okay? Just the whole idea of somebody having blue eyes as opposed to someone having brown eyes. Okay? Different genetics involved here. We'll get into a little bit of these. Okay? Right here. Inherited traits are determined by DNA sequences that comprise genes. The field of genetics investigates how genes confer specific characteristics, okay, that affect health or contribute to our natural variation, our ability there to adapt to the environment, because the environment is always changing. And if our DNA doesn't have the ability to change, we're as good as gone. Okay? We have to be able to adapt. And there's that word again, mutations. They occur when the gene's DNA sequence changes. It can change from environmental factors, like I told you there about those white moths in England during the Industrial Revolution. They were doing great because they were hanging out on the white concrete buildings there. They were having a happy time, but then all of a sudden, all the coal smoke covered all of England. So trees had a black soot. Buildings were all black and dirty and sooty. These white moths were getting pinged off by birds, left, right, and center. And then all of a sudden, one day, there was black moths. The same bug, just a different color. And now, all these black moths are sitting on these black soot-colored trees and buildings, and the birds don't see them. So mutations can be a good thing. Mutations can also be a bad thing. It all depends on your environment. Are you able to adapt? Does this genetic mutation give you an advantage or a hindrance? Okay. Environmental factors there can influence how most genes are expressed. Multifactorial, trait, and height. There are thought to be about 70,000 genes in the human genome, and that is very true. There are thought to be. We're still getting there. These are good. I probably should have introduced these there earlier, but gamete cells, you guys are familiar with. Okay, these sex cells there, they contain half of the DNA. Okay, haploid. And we call half the DNA N because we, me, you, we are all 2N. We have 23 chromosomes from the spermatid, 23 chromosomes from the egg. N plus N gives us 2N. So as a human being, we are all 2N. Okay? Sex cells, gamete cells, are only N. They only have 23. Okay? And that's what 23 chromosomes I'm representing is N, which is a haploid number. Okay? Somatic cells, somatic means body. So this is cells in my arm, my shoulder, my nose, all these kinds of things. These are all somatic cells. Okay? These guys here are all you and me, and they are what we call diploid. Okay? They have 46 chromosomes. 23 and 23, zygote, 46, 2N. N plus N gives us 2N. If there's any issues on that, let me know. But The somatic cells, 46 chromosomes. Gamete cells, 23. We've covered all this. Okay, ovaries, female gametes, ova, ovum. Fertilization, I think we're good to go on that. You remember there that we like the egg to be fertilized within the first third of the fallopian tube, and then it travels on down. Chromosomes, they exist in pairs. Now this is all new stuff coming up here. Chromosomes exist in pairs. There are 23 pairs, right? 23. But there's 23 pairs. One from mom, one from dad, right? 23 pairs, one chromosome from male and one chromosome from female. Okay? So there's 23 chromosomes, 23 pairs of chromosomes in total. 2n. 22 of those are what we call autosomes. These are just non-sex chromosomes, just involved in this and that, and eye color, hair, and all this kind of stuff. There's one pair, it's number 23, that we call the sex chromosomes. Okay? And one comes from mom, one comes from dad, and they could be either X or Y. Okay? There's actually only two. There's double X, and then there's X and Y. Double X means you're female. Okay, X and Y means you're male. Okay. So you have a look here. There's your egg. N is 23. Okay, there's your sperm. 23 chromosomes come together. There's your 46. There's your zygote. Okay. So this just kind of goes into, there's human zygote is diploid, okay, meaning it has two matching homologs there per set, okay. Most organisms are diploid. Normal number of chromosomes is referred to as euploid. So, quote unquote, normal number of chromosomes, 23, is referred to as euploid. There are anomalies though, okay. You can have an extra chromosome. And this causes issues here. Here we have what's called aneuploidy, is the presence of an abnormal number of chromosomes in a cell. For example, a human cell having 45, so one less, or 47, one more chromosome instead of the usual 46, can result in specific diseases there. Like, for example, Down syndrome means having an extra 21st chromosome. And I'll show you quickly, this is what we call a karyotype. And you see, here are the chromosomes of an individual. And you'll see there, each homolog set is made up of 2. So there's 2 sets of chromosomes going on here. That's one pair. That's another pair, and another pair, and you'll see there 22, 23. Okay, and you'll see there that the 23rd, this is your sex chromosome. All right, these ones here are just involved in everything else. These ones here, this one here, determines the sex. I just want to see if I missed anything here. Yeah, it's just saying the same thing. The autosomes are pairs 1 to 22. They carry genes for most traits, but they do not carry the gene that determines sex. Okay? The sex chromosome, to number 23, determines the sex. Female has double X. Male has X and Y. And you'll see there that the female has double X. So it's got, females have 2 X chromosomes on number 23. If there are any diseases within those within those X chromosomes, because there is another X chromosome, females will not exhibit as many genetic type of diseases as males do. Males have the crutch. And what's the crutch? That is the Y chromosome, which I'll show you right here. Just so I'm not talking out my ear here. So you see here, here's what we have, like an X chromosome there with female double X. Okay, so this one here came from mom. This one here came from dad. Dads can have X's, sure, okay, because we're an X and a Y. Moms, they only have the X's there, okay? And you'll see here that there's your X chromosome. Here's your Y. It's not really shaped like a Y in any fashion, but that's what they call it, all right? So this is what defines there for a male. This is called a karyotype. And what this is it just shows you kind of like picture style what chromosomes there are in an individual. Okay, so these are your autosomes here, and this one here. Okay, that's your sex chromosome. The karyotype, just further on, we just saw it there 2 slides ago. All this displays the paired chromosomes used to diagnose genetic disorders. Okay, that's all it is there. And just another shot.
So Ian, just out of curiosity.
Yeah, talk to me. What slide do you want, Laura?
Well, I'm just wondering because you were talking about all the ploidies. Have you ever heard of triploidy?
I have. Talk to me. Do you know something? Triploidy?
Well, yeah.
Go ahead.
So 28 years ago, no, wait, 23 years ago, in 98, I ended up getting pregnant, and I'm bigger now than I was at basically 9 months pregnant with my daughter that I lost, and I had to go through an amnio and everything like that.
Yeah.
And the funny thing is when this happened 23 years ago, triploidy, there was only 5 of us known in Canada that had a pregnancy go that far.
Is it, eh? Interesting.
But now it's more common.
Isn't that interesting, eh?
Yeah. And now it's more common. Like, they've had more, but usually what happens with the triploidy, and I'm not sure, like, I know they tested for Downs and all that.
Sure.
Throughout it all. But normally as a female, your body actually tries to abort. That's why you miscarry within the first few months of. So like when people miscarry within the first 3 months of a pregnancy, it's usually your body is letting it know that there's something wrong.
Sure.
Whereas at the time I was so healthy that I was actually able to carry as far as I did, and then we had to make decisions like because I was told that there was going to be so many defects with her and all of this stuff, and I wanted to donate her organs, but because of the fact that she had like the triploidy, which is she had 69 chromosomes instead of the regular, we weren't able to do that.
I know.
And I was told that there could be a chance that she would be born alive. So I had to make decisions on breathing tubes, everything like that. And I ended up the only tube I told them that would happen if she is born alive is a feeding tube, because there was no way she would be able to survive without the machines anyway.
I know, it's tough,
so it was let's not prolong something that is eventually it's going to end up that way anyway.
So that was 69 chromosomes, eh? So
she had 69.
So that's 23.
Triploidy.
And 23 and 23. And
23.
Yeah. Wow, what a story.
Yeah.
Thank you for sharing. That's yeah.
Well, then because it took 5 years to try again, because you have that fear. When I got pregnant with my daughter, who is now 17, the first 5 months were the tests. You name any possible test that they could do, they did. And I was told in the last test, which I was 5 months, if it came back positive of anything, then I'd have to go for another amnio.
Yeah.
And I'm like, okay, test, come back, be like, everything is good, be everything is good. I don't want another amnio.
Yeah.
I almost had a heart attack with the first one. because my blood pressure, everything, like they had everything there ready for me because of my fear of needles. So it was, but no, when you were talking about all these different like ladies and that, I'm like, okay, let's hear triploidy.
Well, that's what. Hey, you know what, Laura? I just want to say thank you for sharing. And what this is real life. Like you know what I mean. I know that we're showing cartoons here, and you know, but what you just said was awesome, and I thank you so much for sharing. And it must have been such a hard time, but you know what, something there you saw on the screen, you had something to relate about, and this is real life. And where you guys are going, it is real life. And just to share, just a 2-minute little. I also teach a pre-health class, and we use I teach a pre-health statistics class, and I do a case study there every time I teach it. It's on flatulence. You know what I mean? It's on farting, and it can be a tough time trying to get people to understand that, okay, it's not really a comfortable topic to talk about, but you know what? In a lot of ways, the body isn't. The body's kind of gross, you know, and I don't mean that in a bad way. I mean it in a true promise, 100% pinky swear way. And the body does things that sometimes isn't pretty. The body doesn't smell good. These are all things that are real, and you've got to deal with them. And you've got to understand that these things are real, and you might come across them. And where you guys are going to save people's lives. And I have the utmost respect for you guys and where you guys are going. And I'm proud of you guys for this. And you guys have taken the bull by the horns here. And now you've got one more test, and I think the class average will probably crack 80. I don't care. Go for it. And I'm proud of you guys. And thank you, Laura, for sharing. It just kind of, you know, it makes everything kind of full circle now. And I think that was a nice way to close this lecture, which we're almost there anyway. And this is real. And you know, you guys, you nurses, there's a term there you're going to hear that when nurses come home after a 12-hour shift, they need to debrief. They need to let go of what they saw. You work 12 hours in an emergency room, man, you're going to see horrible stuff. People impaled by fences, missing legs, ear bitten off by a dog. I don't know. You guys are going to, you know, where you guys are going, you may not see that kind of stuff, but you know, you will have to debrief at the end of your shift. So when you guys get out there and get working, tell your partner, tell your spouse that there will be times when you'll need to come home and just need to talk at the kitchen table with a cup of coffee and just let it all out. That is for here, for your sense of sanity. Okay, and because this stuff is real, and sometimes it ain't pretty. Okay, thank you so much, Laura. You are 5-star. Thank you. That was aneuploidy. So that is for variances in the number of chromosomes. So Laura was talking there about trisomy. Okay, these kind of triploidy. That's right. And you know what I mean? These kinds of things. They are out there, and you just might see them. Okay. Remember there, the autosomes are 1 to 22. Okay. Sex chromosome is 23. Okay. All chromosomes except Y carry hundreds of genes. This should make sense. Somatic cells contain 2 copies of each autosome and 2 copies of each gene. So somatic cells, they have 2 copies of each of those. Okay. There's our karyotype. Okay, there's our male and female. We go into a little bit more there. We've seen that there, karyotype, genetic art. And you'll see here, there's your male. Okay, so 1 to 22 kind of looks the same. They're not the same, male and female, but you know what I mean. These are your autosomes. And then here's your sex chromosome here. Female there is X. The male has X and Y. Okay, and notice there, it doesn't really look like an X. It's kind of like half an X, right? Okay. Another thing there to know, we've talked about this. Meiosis in males is called spermatogenesis. Meiosis in females is called oogenesis. You guys have seen that. I showed you guys there this slide there earlier. Spermatogenesis starts off with the primary spermicide. Okay, and this guy here is 2N46. It splits, and then right in here, that's where all this genetic variability. One part of a chromosome will jump over to, and it literally, part of a chromosome will jump over to this one, and another part of another chromosome will jump back. So you're switching genes because you want to be prepared for the outside. You want this baby to be prepared for the outside world because the body doesn't even know what the outside world is right now. But it's preparing that baby to be the strongest baby out there. Being prepared for any of the diseases, anything out there. In oogenesis here, it's kind of the same. Here was your primary. This is your primary oocyte. And what happens is it splits, just as the first split here, but it doesn't split into, like this one here went from 1 sperm to 2 sperm to 4 sperm. It doesn't work like that in oogenesis. Okay? It splits, and it produces what's called a polar body. And these polar bodies die producing only, and what they just get discarded. It's just kind of a placeholder. And you see, it splits. This guy divides here again, and these guys are gone. And here, the genetic variability. So this egg that came out this month through ovulation is not the same egg that came out last month for ovulation. Genetic variability. Okay? And this is just an example of 4 spermatids there, right here. And you look at the DNA, gamete 1, gamete 2, gamete 3, gamete 4. They're all different. And that's how the body protects itself against, diseases that are out there. At the conclusion of meiosis, each haploid gamete, should make sense, has one of the 4 chromosomes shown. 2 of the chromosomes are combination. These 2 here. Never before seen before, right? Kind of like a mutation. If it works, then that mutation will carry on to that child's child. If it doesn't work, okay. Modes of inheritance, dominant and recessive, okay? And what we're talking about here, patterns of gene transmission in families are called modes of inheritance, okay? There is a predictability about this, a huge predictability, okay? And it all has to do with dominant and recessive inheritance. A dominant allele masks the presence of a recessive allele. So just allele is just a word there that we're talking about a trait. Okay? So brown eyes. Okay? Brown eyes is what we call a dominant gene. Because you got one gene coming from mom, that's brown eyes. You have one gene coming from dad. And let's say it's a blue recessive. All right? What color will this child's eyes be? Let's do it again. Brown eye gene came from mom. Blue-eyed gene came from dad. What color will this person's eyes be? Brown is dominant. The child's eyes will be brown. But the child, when the child grows up and they have children of their own, they now have a brown gene and a blue gene to pass on to their kids. Let's use another example here. Let's say mom gave a brown brown-eyed gene, and dad gave a brown-eyed gene. So now I got two big brown-eyed dominant. Child's eyes are going to be brown. Okay? Brown, brown, that's the dominant. Let's say mom gave a blue gene and dad gave a brown. Eyes would be brown because brown is dominant. How do we get blue eyes or green eyes or, that kind of a thing? You have a blue gene coming from mom and a blue gene coming from dad. That is the only way somebody gets blue eyes or green eyes or somewhere in there, or hazel. I think hazel's a bit different, but all right. The only way you can see the effects of a recessive gene is you have two of them. Okay? If you have a dominant in there or two dominants in there, that's going to be the dominant. That's what's going to be. But recessive genes, you need two of them in order to see what it is. So a dominant allele masks the expression of a recessive allele. An allele that causes a trait or disease can be recessive or dominant. Now here we were just talking about brown eyes and blue eyes. But we can have diseases. There are genetic diseases out there that work the same. If the recessive gene is the disease, okay? So let's say we get the recessive gene from mom and the dominant gene from dad. Well, this one here is the disease, but it only comes about if I have two recessives. So you see, this is where now the dominant gene covers up the disease gene. So the person has a normal life. They have a great life. They're running around, they're swimming, they're eating, they're doing this. They have no disease. There's no problem. They live to be 99. But if they have kids, because of this genetic variability, we don't know which gene this person gave to their children. And you see, this person, even though they're healthy, they still have that recessive little gene in there, the little devil gene in there. They could be what's called a carrier. So that in the next generation, they could pass along that recessive disease gene. And if mom passes along the same recessive, it doesn't happen very often. That's why these guys, they're still here in human history. They're still playing a part in human history. They've been there for thousands of years. They're still there. They don't come around very often. And we'll get into this more in the next couple slides here. An allele that causes a trait or disease can be recessive or dominant and definitely inherited in either an autosomal or sex-linked manner. Autosomal conditions affect both sexes. X-linked traits mainly affect males. Y-linked traits are only passed from father to son. And you see, it's this one right here that actually makes men or males. And here I removed men because I'm talking about animals as well. This happens in all mammals, all types of vertebrates. Animals there with spines. The males, because of this Y, they can be more of a carrier. And silent carrier, but the gene is still there, so that somewhere along your lineage, there might be a time where a recessive comes in contact with another recessive, and now this person has a debilitating disease. We'll get into these. What do we got here? There's your heredity. Okay, brown is the dominant. So if your mom gave you brown eyes and your dad gave you blue eyes, you got brown eyes. Okay, because the dominant covers up. But if you have two recessive genes here, if mom gave you blue and dad gave you blue, you've definitely got blue. That's the only way to get that type of a phenotype, or what you see. And dominant traits, for example, widow's peak. You'll know that little peak there that comes down. Okay, that is a dominant trait. Freckles, dimples. Even the ability You're going to laugh at this, man. My daughter can't do it, but my son can. And my daughter was really upset. You take your tongue and you just go like that. I apologize. I wasn't trying to be offensive in any manner, but if you can curl your tongue up like that, that's a genetic trait. Some people can, some people can't. Huntington's disease is caused by a delayed action gene associated there with the fourth chromosome. But this is what I want to show you right here. Dominant disorders. So if there's a disease involving the dominant gene, we don't really see those very much anymore, because if the disease is with the dominant gene, the disease is always going to be visible. This person will never make it. Okay? These type of diseases, they only manifest themselves generally with recessive genes. Does that mean that there's not any diseases that are dominant? No. But usually, if a disease is a dominant disorder, it's lethal. And the person usually dies before reproductive age. Okay? Most genetic disorders are inherited as recessive. Albinism, an albino. Okay? That's a recessive gene. And if the squirrel got an albino gene from mom and a regular gene from dad, the squirrel will be a normal looking squirrel. But it will have the ability to carry on that albino gene. Okay? Cystic fibrosis, that's a bad one. Tay-Sachs syndrome, another bad one. Okay? These are recessive genes where if you get one from mom and one from dad, you've got it. Okay? Heterozygotes are carriers who do not express the trait. So a heterozygote is a squirrel that's got a dominant gene and a recessive gene. The dominant gene masks the recessive gene, so it's a healthy squirrel. But it still has the ability to carry that gene on down through its lineage. More about dominance there. You've got dominant gene expresses itself, overshadows the recessive gene. The recessive gene is unexpressed. You don't see it unless it's paired with another recessive gene. But if it's paired with a dominant, you never see it. There's codominant genes. They express a trait in equally. Don't worry about this one. If you guys can knock these two out of the park, that's all I'm good for. Don't worry about codominant. And the last two slides, I thought I'd throw these in there. These are completely unrelated. I don't think I'll test you on sickle cell, but this is the whole idea of an evolutionary plan. Now, what is sickle cell? Sickle cell anemia is this is an RBC. This is a red blood cell, and it looks like a half moon, you know? And surely you're thinking, this can't be good. And it's really not. Okay? It makes people, they can't breathe as well, they can't run long distances, characteristics there with low oxygen levels, because the shape of the red blood cells there, they're completely distorted. It's not like, you can't get enough heme in there. How are you going to get oxygen in there? It's not a great oxygen carrier. And this is, you have a look here. This is called your sickle cell gene. If you have dominant, you have normal blood. Okay? If you have dominant recessive, you have sickle cell trait where you have both sickle cell RBCs and normal RBCs. You can suffer a sickle cell crisis, but you know what I mean? You've got good RBCs in there, you're all right. If you have double recessive sickle cell, now all of your RBCs look like this. Now there's a problem because these guys here cannot carry oxygen at a rate, okay, of normal RBCs. There's problems. But are there benefits to only having these kinds of RBCs. There are, depending upon your geography. Okay? And sickle cell traits provide a survival advantage over people with normal hemoglobin in regions where malaria is endemic. People with sickle cell are unaffected by malaria. So the trait is known to cause significantly fewer deaths due to malaria, especially when the Plasmodium falciparum is the organism. Okay, so this is the bug there that's causing malaria from the mosquito bite. And if you've got sickle cell, you're not affected as much. So there are genetic deficiencies that really aren't. It's all based on geography. And maybe a million years ago, there was a mutation there. This person got sickle cell, double recessive. This person survived while all the other people didn't. With normal red blood cells, the malaria got them a million years ago, and this person was the only person left. Him and, you know what I mean? He got a spouse. There you go. They all have sickle cell, and they're all alive to this day because of the sickle cell. So that's what I got. That's all I got. And I just want to say that proud of you guys. You guys just keep on trucking. You guys have a week. Your test will open. I'm thinking the same. I don't know, 8 o'clock till midnight. That's what I'm thinking. Get it over with in one day, and you guys will have lots of time to do it. It'll be about 100 questions somewhere. I'm making the test right now. It's done. I'm just trimming off some stuff and cleaning it all up. But I hope you guys are well. You guys are doing really great. Just keep at it and keep in touch, man. You guys got any questions?
Thank you so much for everything, Ian.
Oh, you